Welcome everyone to this webinar regarding conception. My name is Hansson. I'm Senior Professor of Biomedical Ethics at Uppsala University, and I'm leading one of the work packages in conception, work package 4, related to European breast milk collection procedures. I'm co-leading it with Marie Tile from the company UCB. So just for your information, the basic background, the basic rationale for conception as a whole is to provide more evidence for drug treatment during pregnancy and at breastfeeding for women. So there is a well-recognized and well-acknowledged problem that we don't have sufficient amount of evidence in order to provide women with a clear guidance on when to take a drug and when to not take a drug, and also when recommended to breastfeed or not breastfeed. So I will provide an overview about this project, and then the presentations will follow on that. So welcome everyone. These are the people that you will meet. I've already introduced myself. Andrea Wutte is the head of BBMRI-ERIC services on quality management, and BBMRI-ERIC, she will shortly present that, is a Europe-wide research infrastructure for biobanking and biomolecular resources. And then you will meet Associate Professor Pawel Baranczewski, who is at Uppsala University at the Department of Pharmacy, and is going to present what they have done regarding the bioanalysis of requirements for demonstration studies. And then you will also meet Professor Hedvig Nordeng from the University of Oslo, who has done a lot of lactation studies during the years, and she has been responsible for one of the very successful stories from the conception project, the work package 4, that is a lactation study on levocetirizine levels in human milk, part of allergy treatment, as some of you may know. So this is the group level with Daniela Krasser from BBMRI-ERIC is going to host it, and also we have people around in communication and in administration. The mission for conception is to build and test out in detail a trustworthy pan-European ecosystem for generating, monitoring, and providing robust and rapid evidence on medication safety in pregnancy and breastfeeding. As I started to say, there is a great problem that we today worldwide don't have sufficient evidence in order to provide proper guidance to doctors and to women regarding medication during pregnancy and breastfeeding. So the conception project focused on different aspects, and we have several work packages. I will just focus here on some of those related to the more scientific content. The first work package is trying to take the stand from the fact that we have pregnancy registries around the world, but they are of insufficient quality and they don't cover enough data. And there are no standards with regards to data and data quality. So the important thing here for work package 1 is to make sure that we have available clinically actionable and timely evidence to patients and healthcare professionals via the medication label, so that all information is already included in the label of the specific drug. It's also important to reduce inconsistencies in format and content between different information sources, so that we can speak with one voice to women and to the healthcare professionals regarding label. So the idea to move the medication labels closer to become a gold standard of information and pregnancy and lactation across treatments. So this is the place where you go. So that's one of the important first work packages. The second work package of ConcePTION is related to development and use of innovative analytical approaches. So here the idea and the task is to develop, to optimize, and also validate methods to search adverse event data sets for teratogenic signals. There is already a lot of things being done in this, but this is the idea to really optimize and validate these methods, that we have a uniform way to search and to get teratogenic signals regarding specific drugs. And also to improve quality and collation of phenotypic information with possible and likely teratogenic syndrome. This is a field that can be as big as one wants, but it is important to be able to collect and interpret this phenotypic information in a proper way. So this is the work package 2. The work package 3 is also working very much related to the content. So the idea is here to provide predictive preclinical models for different drugs. And they are using different means in order to develop that, this sort of issue of predictivity. In vitro lactation studies, where they can investigate the properties, the pharmacokinetic properties, how drugs behave in different organs and in different contexts. They're also doing in vivo experiments in lactating non-clinical species, and these are in this project is about small pigs, mini pigs that they use. So they provide an array of information that can be used to predict what is going to happen if we use this drug. And then we come to work package 4, and that is the work package that we will describe in more detail in this webinar. The idea is here also, in addition to what is done in the other work package, to build a sustainable Europe-wide collection of breast milk for research. There are, of course, Europe-wide or very large collection of breast milk, but not for research purposes often, but for clinical purposes, to be able to provide breast milk for women that needs them. But here the idea is to do it for research purposes, and there are, of course, biobanks around Europe that have been built and collected in order to do research. But they are done in different ways. One collect breast milk in different ways, one ship them, one store them in different temperature in different ways. So we need to have standardized procedures so that we can collect and ship and store breast milk across Europe in a way that the data that comes out of the analysis later can really be trustworthy and of great validity. The other part is related to what Pawel will tell you more about, and that is the development of standard operation procedures for bioanalytics, stability tests for demonstration drugs. We are doing 5 demonstration projects, as we'll shortly tell you, and we need to know that when you ship breast milk collected, for example, in Norway, and store it in the biobank in Sweden, you need to know that the drug compound is stable under the condition that we use for the transportation and the shipment, or if it comes from France, from Switzerland in the same way. So we need to have stability tests for the rotation. We need to be sure that what we can deliver is of utmost quality. What we have done also already, we have done these standard operation procedures for collection, for procedures for bioanalytics, and we also developed an umbrella protocol and also specific protocols for 5 different demonstration studies. And the idea is here to have a proof of concept that if you do it in this way, you collect in this way, and you can do the analysis according to these standards and these procedures, we can do it for different kinds of compounds in different disease contexts, and we can show that it works. The challenge is often to do recruitment in a proper way, to find the women that are willing to participate in the demonstration study. It's not a case that they are unwilling. They are often very, very eager to participate. I think Hedvig will tell you more about that from the Norwegian study. But we need to make sure that all these components, elements of lactation study actually works in practice. We need to have standard documents for pre-examination process, breast milk handling, and biobank, and that has been developed. That is also together with the quality management structure for biobanking and pre-analytical handling of human breast milk specimen. That is very much what Andrea Wutte from BBMRI-ERIC will tell you about. So these are the key outputs to provide systems, procedures, ways to do it for a lactation study and to biobank breast milk samples, and in case that is appropriate, with associated blood from the woman and eventually also from the child. The basic requirement here that we need to fulfill all the way through, and you remember we talked about the label of the drug to be the gold standard. This is where you go there in order to get the appropriate and validated information. We need to make sure that what we come up with meet regulatory standards. The way we do it, the way we analyze, that there are fulfillment of all regulatory standards. These are currently issued by the Food and Drug Administration of the United States, FDA, but also EMA has the regulatory standards. So the results that we come up with, they must be fit for label, and that is the primary requirement. That has been the guiding idea all the way through, and will be. So our way of selecting laboratory facilities and how analysis is done according to the book, and that is very important. So the demonstration studies are 5. I will not go through them in any detail here, but they will result in operational analytical standards to conduct lactation studies in Europe. So one of the studies on allergy treatment levocetirizine is approved and finished. It's going on in Oslo, and they have already collected the samples. Venlafaxine is ongoing in Lausanne. Metformin and prednisolone are in development. So we have protocols ready, but we are sending that out now in all over Sweden. Amoxicillin is approved and ongoing. So these are demonstration data that are currently going on in different countries with different drugs. We are facing key challenges, and as I said, the recruitment of women is one of those challenges. In Sweden, we realized for our demonstration studies that when we ask clinicians if they prescribe these particular drugs to the patients, to the women during pregnancy and association with breastfeeding, some said, no, we don't do that. And some said, yes, we do that. So with this conflicting information, we went to the National Board of Health and Welfare and asked them to do a match of the prescriptions registry, the National Prescription Registry and the Medical Birth Registry in Sweden, and found a match of cases when you actually did prescribe that. So that is what we are using. And it's a kind of a natural experiment in that sense going on. These are prescribed on sometimes not sufficient evidence according to the scientific standards. And we are going to see how that works. So we are not prescribing any drugs. These are done by the doctors already, but we are following it and observing. Key challenges has also been to manage logistics, to be able to ship them in a way that you have a stable compound at the end. And I think there will be more examples from Norway later regarding challenges and regarding bioanalytics. So this is what I've already said. We are part of the ConcePTION project. It has received funding from the Innovative Medicines Initiative number 2, IMI, now moving on to Innovative Health Initiative, but the support here today is from the IMI. They provide funding to the academic partners, the public partners, and the pharmaceutical industry participates with equal amount in kind contributions. So with those words, I think I leave over to Andrea.
Yes, thank you, Mats. And thank you to the organizers, to the guests and the ConcePTION colleagues. Thank you for the invitation to present this overview of activities that BBMRI-ERIC Quality Department team is proudly contributing to the ConcePTION project. Just briefly, and Mats mentioned it already, the description of BBMRI-ERIC. So we are a very large European research infrastructure. We have 24 member and observer countries across Europe and including international organization WHO, IAC, and we all focusing primarily on biobanking and on biomedical research based on human specimens. So one of our key mission is within BBMRI, and I think that is why we are also contributing to this project, is the harmonization, standardization of biobanking activities to mainly ensure the interoperability in biomedical research on human samples of any disease classification, but always with the primary goal of advancing research for the benefit of human health. And we have this always in our mind. So our infrastructure counts more than 900 biobanks and most important, including their research groups. So the research actors are within the biobank. So there is a big knowledge on biobanking activities, but also on different disease types. So we enabling and harmonizing research on human specimens. And this is one of the key priorities within our biobanking community. So I think you don't see my, now you see my second slide, yes. So therefore it is, of course, also for us as BBMRI-ERIC research infrastructure, very important to see new collections in our domain, let's say that way, and collecting breast milk and associated blood samples and medical data is something that we welcome in our biobanking world. So we contribute here in this project, and Mats already mentioned it, that we are developing commonly a harmonized and standardized sample collection procedure of breast milk and blood samples in the context of this work package 4. And what we want to is to provide a lot of standardized templates for future sample collections of breast milk for more collection sites to come in the future. The objective that we face in work package 4, and that is that we review and verification of key processes and methods within the pre-analytical processes that are needed to collect samples. Then we have a group of technical experts who review the standardization activities. We design or we started, and we're almost there, design a basic structure for a quality management system from a set of applicable European and international standards and guidelines that are necessary to perform such studies or biobanking activities, and development of standard documents for pre-examination processes. And here, as mentioned already, the collection, stabilization, transport, storage, processing, analytical testing for breast milk handling and for blood. In this sense, we see biobanking activities as part of an overarching workflow in which interdisciplinary and international teams are working together and coordinating their processes to generate quality assured and scientifically sound result. And this is a challenge, and we have taken this up in work group 4, and we see on one hand the clinical process for patient care, clinical trials and biobanking processes are very closely related, but differ in some aspects. And they differ in some aspects regarding regulatories, requirements, standards, guidelines that have to be followed. So therefore, it is of utmost importance to incorporate the requirements in the entire workflow. And we try to take this into account with the documents and system that we set up, always in the light when in future sample collection take place and milk collections or are collected in more clinical sites, that we provide the basic structure so that we can harmonize these procedures also for the future. When we show you what the organization of a European breast milk collection can or should look like, we see that this quality management system and the system of collection is based on these regulatory guidelines and standards that are available and are incorporated in the documentation that we are preparing and that we have prepared already within ConcePTION. So you see a lot of the basic ground, which is, as you see here, ISO 23887, a number. It's an international standard that defines the biobanking general requirements, but it also has the future perspective that a biobank can be accredited accordingly. Then a lot of other standards, international standards that are relevant, pre-analytical standards that describe sample processing with a specific intended use of analytical methods, general requirements for quality management, ICH, GCB guidelines, the guideline of the FDA, ISO 15189, a standard for medical laboratories. All these aspects have been put into the standardized procedures, into protocols, and will also be visible then in the reports that come out of the study. So as Mats already mentioned, 5 demonstration studies have been initiated already to collect based on documents that the whole work package 4 team has commonly created. Now I think I can say that all these documents are used to go from these demonstration studies into the storage facility to the analytical center in Uppsala, and then the output, the clinical data and results from the analysis are made available for any prediction of or for therapeutic use. When I just want to show you a little bit more briefly how we are looking at the biobanking standard, and here the process requirements we took a closer look at. The description, the international standard is a document that is harmonized in the International Organization of Standardization by worldwide member states of the ISO platform. And the standard was developed in 5 years and came to a good product that we can now use to define quality aspects within biobanking activities. And as you see, the collection of biological material, reception, distribution, transport, traceability of the material and the data, the preparation, quality control, and validation as one of the key aspects. All these bullet points have a more granular explanation how a biobank should work accordingly. When we go to the next slide, this is what we did. So preparing for sample collection, processing, transport, sample storing, sample analysis, quality control aspects in the work package for with all the partners contributing. Process description, SOPs, forms, and checklists, questionnaires, which will at the end lead to a file system that is then handled within the IT in a laboratory information system or in a biobank information system. And we have here some examples of standard documents so that you just get a short overview how these documents look like. We can, of course, coming from the ICH-TCB guidelines, the information to the participation or to the mother who is participating in a study, informed consent information. Then we have templates for the procedures for collecting and processing breast milk samples. And we have also these procedures described for collecting and processing blood samples, and questionnaire for mothers, questionnaire for the clinical site, and questionnaire for the blood sampling on the health status of the patient, work instruction for sample shipment, then sample data files that we can characterize the samples, but also how to work with encrypted data, sample storage conditions. So these are just examples of what we have done together in a great collaboration within the work package for, and of course, we are not at the end of the conception. There is a lot for a lot of next steps to be done, but we are really looking forward to those steps. And with this, I would like to hand over to Pawel.
Thank you. Good afternoon, everyone. My name is Pawel Baranczewski, and I'm working at the Department of Pharmacy at Uppsala University in Sweden. I'm also part of Swedish SciLife Lab Laboratory Drug Discovery and Development Platform, and I would like to briefly update you on bioanalysis requirements for the demonstration study within IMI conception project. So as introduction, Mats already mentioned, we are aiming to perform 5 demonstration studies, and this will be sort of proof of concept in terms of estimation risk and for during lactation process for different drugs. So 5 studies will be performed or are ongoing at different countries to have approach also difference between countries. One study is in Switzerland. In Oslo, it's already finalized, and in Toulouse, amoxicillin, and 2 studies will be performed in Sweden. And most of the bioanalysis will take place here in Uppsala, but amoxicillin samples will be also analyzed in France. So we have collaboration with other working packages within IMI conception. And after discussion, we decide to run bioanalysis using liquid chromatography mass spectroscopy technique, which is robust, sensitive, specific, and we can analyze different type of analytes, different type of compounds. So once we have this method, bioanalysis, we can quantify compounds in breast milk. And of course, this data we will use to describe disposition of different drugs within mother population. Also, aim is to use population PK modeling to identify different covariants which contributing to population variability. Of course, this data will be downloaded to ConcePTION database, and on end, best practice subdocuments will be shared, published, open for everyone. Saying this, for bioanalysis, we decided to make as good as we can, and therefore we try to apply all regulatory authorities, FDA, EMA, guidelines and recommendation. We did also have very good consultancy with Dr. John Smiraglia from USB Pharma Company to apply even pharma companies' standards. And aim of this all work was to establish best practice and operating standard procedures for analysis method and compounds in human breast milk. For every method, for every compound, we prepared study validation plan where we describe all specific issue related to compounds, use instrument and analysis, and all results of validation process will be reported in study validation report. And here we will discuss how develop and validate method fulfill all parameters and criteria. And on this slide, you can see matrices which we use. It's mostly human breast milk, of course, but also human plasma. We were interested. And later you see validation parameters, reference standards, criteria, critical reagents, calibration curve, quality control samples, selectivity, specificity, sensitivity, and accuracy was very strict, and we wanted here to follow regulatory recommendation and criteria. We also spent quite much time on testing stability of the analytes in different matrices, and I will briefly refer to this later. And on the right side, as you see, you can see the first side of study plans and study report from Uppsala and also one from Toulouse. And as sustainability, we already summarized and described our experience with method development for 4 compounds in form of manuscript. And this manuscript was then submitted to Journal of Chromatography and currently is under review process. And of course, very briefly about workflow for this all bioanalysis. First, of course, we have sample handling. Samples are delivered to biobank and from biobank to our laboratory. We have this sample preparation process, which is quite fast, but needed some adjustment for different compounds. As I mentioned, bioanalysis are performed with UPLC mass spectrometry, and here you have picture of instrument on which we perform the analysis. And finally, yes, you need to take care about results, reporting, and storage of the results. And yes, if you talk about bioanalysis, you always need to show some representative chromatograms, and here we have how it's looked like for 3 of the compounds. cetirizine, venlafaxine, and active metabolites or desmethylvenlafaxine. As you see, the peaks are distinguished. We don't have any major background, and even if it's a very low concentration here, we see also very good separation between peaks, so we don't have carryover and so on. And finally, I want to show you the results. And here we have the first slides, which summarize results of this BBMRI method validation for cetirizine to the left and venlafaxine and the active metabolite to the right. And here you see calibration range, calibration curve, concentration, which we can detect, accuracy and precision. And as you see, all these parameters are within 15% deviation, which is accepted by regulatory authorities. Also, I wanted to stress that we actually aim it for as sensitive methods as possible. So we wanted really to detect all possible low concentration in breast milk and have control. So the methods are really sensitive. And also, as I mentioned, we spent quite much time on investigating actually stability of our compounds in breast milk over different time. And this was at room temperature, 4-degree temperature, at freeze temperature, and also so stability for 5 cycles. This was mostly to ensure that nothing happened with compound during sample taking, during transport, and during sample handling process. And on the next slide, we have exactly the same results for metformin in human plasma to the left and in human breast milk to the right. Also, as you see, in this case, we didn't have any major variability. We have very sensitive methods. And also in this case, we perform stability test in different matrices and over different time, just to ensure that nothing happened to the samples. And saying this, I would like to thank you all work package for bioanalytical team members, of course, IMI ConcePTION friends and colleagues. And thank you very much for your attention. And after me, it will be Hedvig Nordeng talking about cetirizine levels in human breast milk. Thank you very much.
Thank you very much. So I will start sharing my slides. I hope you can see my slides in full screen modus. So first of all, thank you to the organizers for the invitation. It is a pleasure to be here today to talk about one of the demonstration studies in work package 7, the levocetirizine human lactation study. So what I will do quite briefly, give a very short background. I will focus on the design of the study, the sampling regimen we landed on. I'll touch upon some of the discussions we had with the ethics committee and what were their concerns, because I think that's interesting shared learning. And I will address some of the practical issues we have or challenges we had to overcome, say a little bit about the recruitment strategy, and then I will give you some results. And I will have to do all of this in 10 minutes, which is very short. So I will be very brief with the background. The policy from the regulator's standpoint is that human lactation studies should be performed when drugs will be used by breastfeeding women. The European Medicines Agency have revised their guidelines with really wanting to step up and wanting us to do better than we currently are, because we know that human lactation studies are rare and that often the drug labels are not updated, even when there are studies being performed. Now, for us, choosing levocetirizine was felt kind of natural. We know that allergy is one of the most common chronic conditions in women of reproductive age and also among women that are breastfeeding. And we know that second-generation antihistamines are the first-line therapy. However, so despite the number of breastfeeding women using antihistamines, the data is surprisingly little. And here you can see on the slide, for levocetirizine worldwide, we only had information from 3 women, and they provided 1 sample each, and this was not at steady state. So this was the compound we decided to study. And we had to answer and discuss so many questions. How many women should we include? How much milk should we ask from every one of them? Should we ask for a full breastfeed expression, or would it be sufficient with a mid-sample? We know that the foremilk and the hindmilk have different concentrations of fat, so it does have an impact on the concentration of milk. How many samples could we ask from each woman? Do we use an intensive day sampling or just random sampling? Do we ask for a milk-only study, or do we ask for a milk and blood? How do we set up the assay? And I think Pawel has given a very nice example of that. How do we ship the samples? How do we store it? And that we have heard a very nice presentation also from the biobank. And then how do we do the calculations? Do we do the more traditional pharmacokinetic calculation of the relative dose, or do we try to do more advanced population-based pharmacokinetic modeling? So these were the kind of questions we were asking ourselves. And all of them, well, we had to take some pragmatic decisions. We landed on a target sample size of 25 women and a milk-only study, which is regulatory acceptable. And we really wanted to tailor it to the women's needs and to make it as non-invasive as possible. And therefore, we wanted them to be able to provide the pump the breast milk samples and store them in their own home freezer to express all the samples and then send them directly to the biobank in Uppsala. We landed on 20 milliliters. In the San Diego biobank, they are asking for more, up to 60 milliliters, but we thought that 20 milliliters is sufficient. And we designed the study with 1 day of intensive sampling. So we asked the women to provide breast milk samples at times 0, then at 2, 4, 6, 12, and 24. And then at 3 other days, we said, well, give us a random sample at any time. You just need to be very specific that you note the time point when you take the drug and the time point when you express the milk sample. And of course, they were shipped and analyzed by our colleagues in Sweden. And this slide just shows you really how the study was designed. We recruited women through social media, and everything was signed online. So it was an informed consent, electronic informed consent. They filled in a baseline questionnaire with information about health characteristics, maternal and child characteristics. And at that time point, we called them up and we had a startup call where we explained all the details and answered any questions they might have. And at that time point, if they were still motivated and willing, well, at that time point, we sent them all the sampling equipment that they needed, and we also provided them with a breast milk pump. All women had to be taking levocetirizine for at least 2 days, and we needed them to be at steady state level. And that's where then they started to provide the samples, and And every day that they took a sample, well, they also filled in a follow-up questionnaire. And at these questionnaires, there were also questions about how the child was doing. Here is just some of the information, or what we landed after discussion with the ethical committee. First of all, they required some justifications for some of the data that we did collect, because it is sensitive information. They required a specific informed consent from the father, because we were asking also follow-up questions about how the child was doing. So we added that. In the main informed consent, they actually wanted us to provide a little bit more information about the conception project in general and how the data would be shared. And they asked for a specific informed consent regarding the storage of the data in the biobank. And we had to also clearly specify that no predictive genetic research would be performed. Now, this was legislation that applied to Norway, and in different countries there are different ethics committee might have different comments or concerns. Here's just a slide showing some of the practical issues. We opted for milk bags because women were familiar with them. And we also tested the equipment, and we tested the shipment, so we know exactly how many days it would take to come to Sweden, and we landed on shipment on Mondays and Tuesdays because we didn't want them to get stuck in the customs. And here's just a slide showing the recruitment strategy. We had help from the Conception Consortium to make a recruitment video. We had great support from a patients organization in Norway, not only the allergy patients or asthma and allergy patient organization, but also women's health organization, lactational support organization. We developed a website for the study. We posted banners on social media, and we even made a small cartoon. And we saw that it worked really well. So remember that our target sample was 25 women. We had more women wanting to participate than that target, and we didn't want to say no. So in total, we included 37 women because we weren't actually sure whether all of them would be able to provide us with the 9 samples each. Some of them we knew had not a lot of milk, and they were really, some of them were in their early stages of lactation, and some were in later stages and had maybe a child up to over a year. We did require that all the women the child had to be at least 1 month old, and that the breastfeeding had to be well established because we didn't want to disturb the mother and child that first months. So 28 women were able to provide us with 9 samples, and you see here in total what they provided. First day was the intensive fixed sampling, and then 3 other days the random sampling. And all of these samples were shipped and then aliquoted into over 4,500 aliquots that were stored, and some of them then analyzed. And just to show some of the results. So, and these are thanks to our colleagues at the Uppsala University, showed mean concentration of 12 nanograms per milliliters, and of course a quite broad concentration range depending on sampling time. So this is what we're currently analyzing, and we've been cleaning the data files and writing up the manuscript. So just to summarize, what have we learned? The women are very eager to participate, to donate their breast milk when they're already using these drugs, and many of them tell us that they have been struggling to find information, not only when they're breastfeeding, but many of them have been on these drugs in pregnancy as well. Pumping at home worked very well for them. They didn't need to come into any of the hospitals, and then of course we could recruit women from all over Norway, not only in Oslo where I'm situated. Social media worked well, the electronic questionnaires worked well. We did have a few of the women having some punching errors. And so we think that one possible improvement is to use QR codes and have them more use mobile phones to take pictures and scan. We saw that it was important to have a close follow-up, reminders, being available at all times to ask them, for them to ask us questions, and of course the piloting was important. Yes, and I mentioned the QR codes, and yes, there's a lot of paperwork to ship over across borders, and so we had to do. It was a bit of a struggle in the beginning, but in the end, well, in the beginning we wanted the women themselves to order the DHL pickup, but we found out that that was too many questions, so we landed on ordering everything for them ourselves. And with that, I would just again like to acknowledge my colleagues. It's really been a very nice learning experience of how we can work together across European countries and to do these kind of human lactation studies. So thank you for your attention.
So thank you all for participating and for your interest and engagement. We who are running this study are of course available. You may find us and have contact with us through the ordinary means of mail or whatever. So thank you for participating. Thank you. I want to thank my colleagues for doing the excellent presentations and for the ConcePTION and the IMI to make all this possible. So with those words, I thank you and I wish you a good continuation of the day or a good evening.